First, we are discussing about a prion is a type of infectious protein that can cause neurodegenerative diseases in humans and other animals.
The term prion stands for proteinaceous infectious particle. Unlike typical infectious agents such as bacteria or viruses, prions do not contain DNA or RNA. Instead, they are misfolded versions of normal cellular proteins that have the ability to induce other normally folded versions of the same protein to adopt the abnormal, disease-causing conformation. Prion diseases, also known as transmissible spongiform encephalopathies, TSEs, are characterized by the accumulation of these misfolded prion proteins in the brain and nervous system. This accumulation leads to the formation of insoluble aggregates that disrupt normal cellular function, resulting in the progressive degeneration of brain tissue. Some well-known prion diseases include Creutzfeldt-Jakob disease, CJD, in humans, mad cow disease, bovine spongiform encephalopathy or BSE, in cattle, and scrapie in sheep. These diseases are typically fatal and have no known cure. Prions are notable for their ability to transmit their abnormal conformation from one protein molecule to another, effectively propagating the disease without the need for genetic material like DNA or RNA. This unique mode of transmission and the complex nature of prion diseases have led to ongoing research to better understand their mechanisms and develop potential treatments. Second, we are discuss about viroids. Viroids are small, infectious agents that are even simpler in structure than viruses. Unlike viruses, viroids lack a protein coat and consist solely of a short, single-stranded RNA molecule. This RNA molecule is usually only a few hundred nucleotides long and is capable of causing disease in plants. Viroids were first discovered in the 1970s and are known to cause various plant diseases by interfering with normal plant cellular processes. They do this by using their RNA to disrupt gene expression and manipulate the host plant's metabolism. Viroid infections can result in stunted growth, abnormal development, and reduced crop yield in agricultural plants. Because viroids lack the protein coat found in viruses, they are much simpler in structure and function. They do not encode any proteins themselves. Instead, they rely on the cellular machinery of the host plant to replicate their RNA. Viroids are typically spread from plant to plant through mechanical transmission, such as when plants are wounded or when contaminated pruning tools are used on healthy plants. There are several different types of viroids, each causing distinct plant diseases. Some well-known examples include the potato spindle tuber viroid, PSTVD, which affects potatoes, and the citrus exocotis viroid, CEVD, which affects citrus plants. Viroids are an interesting area of study in the field of molecular biology and plant pathology because of their minimalistic structure and ability to cause disease without the typical genetic material or protein components found in traditional pathogens like viruses or bacteria. Third concept is viruses, about which we can say biological viruses are tiny infectious agents composed of genetic material, either DNA or RNA, surrounded by a protein coat. They lack cellular structures and can only replicate inside living host cells. Viruses hijack the host's machinery to reproduce, leading to various diseases in animals, plants, and microbes. They play a crucial role in the spread of illnesses like the common cold, influenza, HIV, and COVID-19. Now we can learn about the mycoplasma. Mycoplasma are bacteria that lack a cell wall, giving them a unique and flexible shape. They are among the smallest and simplest self-replicating organisms. Mycoplasma species can cause various infections in humans, animals, and plants due to their ability to attach to host cells and interfere with their function. They are known for causing respiratory and urinary tract infections, as well as other illnesses. Archaebacteria, often simply called archaea, are a group of microorganisms distinct from bacteria and eukaryotes. They represent one of the three main domains of life, alongside bacteria and eukaryotes. Archaea are known for thriving in extreme environments such as hot springs, acidic lakes, and deep-sea hydrothermal vents, leading to their initial classification as extremophiles. Their genetic and biochemical characteristics set them apart from bacteria and align them more closely with eukaryotes in some aspects. Archaebacteria have unique cell membrane structures, often composed of isoprenoid lipids, which provide stability under extreme conditions. They can be classified into three main groups: halophiles, salt-loving; thermophiles, heat-loving; and methanogens, methane-producing. Methanogens are crucial for the biogeochemical cycling of carbon and play a role in anaerobic environments like the digestive tracts of some animals and certain wetlands. Research into archaea has revealed insights into the evolution of life on Earth and the diversity of biological processes. They have potential applications in biotechnology, such as in the development of extremozymes for industrial processes. Overall, archaea represent a fascinating branch of life that continues to intrigue scientists exploring the fundamental aspects of biology and the possibilities of life in extreme conditions. Now about the cyanobacteria. Cyanobacteria, often called blue-green algae, are a group of photosynthetic microorganisms. They played a significant role in Earth's history by producing oxygen through photosynthesis billions of years ago, shaping the planet's atmosphere. These bacteria are found in various environments, from oceans to freshwater bodies, and even in symbiotic relationships with plants and fungi. Some cyanobacteria can form colonies and produce toxins harmful to aquatic life and humans in nutrient-rich water, leading to harmful algal blooms. Despite potential hazards, cyanobacteria contribute to nutrient cycling, carbon fixation, and are studied for their potential in biofuel production and environmental remediation. Generally we can say the bacteria or the eubacteria, commonly referred to as bacteria, are a diverse group of single-celled microorganisms. They belong to one of the three main domains of life and are characterized by their prokaryotic cell structure lacking a nucleus. Eubacteria are found virtually everywhere on Earth, displaying remarkable adaptability to various environments. They play essential roles in nutrient cycling, some causing diseases, while others are beneficial, aiding in digestion and environmental processes. Their genetic diversity and rapid reproduction make them subjects of extensive scientific research, offering insights into genetics, evolution, and potential applications in biotechnology and medicine.